Here's a summary of the entire biology paper 2 spec. First, we have homeostasis, which is defined as the maintenance of a stable internal environment. This just means keeping the conditions in your body just right for the cells to work the best. This could be things such as body temperature, blood glucose levels, and the water content of the body. These conditions are kept constant by using nervous and hormonal responses. Nervous responses involve the nervous system, which allows humans to react to their surroundings and coordinate their behavior. It's made up of the central nervous system, which includes the brain and spinal cord, and this acts as a coordination center. It also contains receptors which sense a change in the environment, which we call a stimulus, and effectors, which can be a muscle that contracts or a gland that secretes hormones. Everything in the nervous system is connected by neurons, which transfer information in the form of electrical impulses. There are sensory neurons that connect receptors to the central nervous system, motor neurons that connect effectors to the central nervous system, and relay neurons that connect the sensory neurons directly to motor neurons during a reflex action. You can have two types of responses: voluntary responses, where the impulses go to the brain and the brain coordinates a response, and reflex responses, where the information doesn't go to the brain, instead goes to relay neurons in the spinal cord. These are what act as a coordinator. The route that these reflex responses take is known as the reflex arc. Reflex responses are rapid and automatic, which is useful as it can protect a person from injury. Gaps between neurons are known as synapses, and here the impulses move across the gaps in the form of neurotransmitters, and they move across by diffusion. The endocrine system also transfers messages around the body, but these are in the form of chemical messengers known as hormones instead of electrical impulses. These travel around the body in the blood instead of neurons. Hormonal responses are slower than nervous responses, but the effects are longer lasting. The endocrine system has 6 glands you need to know, and these are the pituitary gland, thyroid gland, adrenal gland, pancreas, ovaries, and testes. The adrenal gland produces the hormone adrenaline, which prepares the body for fight or flight during times of stress or fear. It does this by increasing the heart rate and boosting the delivery of oxygen and glucose to the brain and muscles. The thyroid gland produces the hormone thyroxine, which controls the basal metabolic rate. This is important for growth and development. The levels of thyroxine are controlled by negative feedback control, which stops the gland from producing too much or too little hormones. The pancreas produces the hormones insulin and glucagon, which are both important in controlling blood glucose concentration. If blood glucose concentration is too high, the pancreas produces insulin, which converts the glucose into glycogen and stores it in the muscles and liver. If blood glucose is too low, it produces glucagon instead, which converts the glycogen in the liver and muscles back into glucose and releases it into the blood. Just like thyroxine, both of these hormones work using a negative feedback cycle to keep glucose levels constant. When this mechanism doesn't work properly, the person will not be able to control their blood glucose levels, which means they have diabetes. There are 2 types of diabetes. The first is type 1 diabetes, which is when the pancreas produces little to no insulin. This means their glucose levels remain high. It is inherited and can be managed with insulin injections, specifically around meal times. Type 2 diabetes, on the other hand, is when the body cells become resistant to insulin. This means the pancreas still produces insulin, but it doesn't cause the blood glucose levels to decrease. Insulin injections won't work with this, so the best way to treat it is by having a carbohydrate controlled diet and by doing regular exercise. Obesity is a risk factor for type 2 diabetes, which just means that an obese person is more likely to get it. Next, we have testes, which produce the hormone testosterone in men. This produces sperm and causes physical changes such as facial hair growth. In women, however, the ovaries produce estrogen, which matures the egg cells at puberty and causes physical changes such as breast development. It also plays a vital role in the menstrual cycle along with 3 other hormones. Estrogen causes the uterus lining to thicken and grow and stimulates the release of LH and inhibits, which means to stop, FSH. LH is known as luteinizing hormone and is produced by the pituitary gland. It triggers the release of an egg, which is known as ovulation. FSH is also produced by the pituitary gland, and this causes the eggs to mature in the ovaries. It also stimulates estrogen to be produced. The 4th hormone involved is progesterone, which is produced in the ovaries. It maintains the uterus lining and inhibits both FSH and LH. The menstrual cycle lasts 28 days, and days 1 to 4 is when menstruation occurs. This is where the uterus lining breaks down. From days 4 to 14, the uterus lining rebuilds and the egg develops in preparation to being released. On day 14, ovulation occurs, and this is when the egg is released from the ovaries. Then from days 14 to 28, the thickness of the uterus lining is maintained, and if no sperm fertilizes the egg by this point, the cycle starts from the beginning again. Here are 2 graphs that show how the levels of these 4 hormones change throughout the 28-day cycle. Next, we have contraception methods, which are ways to reduce fertility and prevent pregnancy. These methods can be hormonal or non-hormonal. Hormonal methods use the hormone estrogen, as this inhibits the release of FSH, which stops the egg from developing and releasing. They also produce progesterone, which causes the cervix to produce thick mucus, which stops the sperm from passing through. Here's a summary of all the hormonal methods you could be asked, and here are all the non-hormonal methods you could be asked. Some people find it hard to get pregnant, so can take fertility drugs to increase their chances. These contain the hormones FSH and LH, which can stimulate ovulation. However, if too many eggs are stimulated, it could lead to multiple pregnancies. IVF or in vitro fertilization can also be used to increase chances of pregnancy. The process starts with the woman taking the fertility drug with FSH and LH to stimulate several eggs. The eggs are then collected and fertilized with a man's sperm in a lab. They then develop into embryos, and when they're tiny balls of cells, they're inserted into the mother's uterus. This does increase the chance of a woman getting pregnant, however it does have a low success rate and is both emotionally and physically stressful. There's also a potential for multiple births, such as twins and triplets or even more, which can increase the chances of miscarriage. Now for topic 6, the first part is on the difference between sexual and asexual reproduction. Sexual reproduction involves two parents, where each parent contribute gametes, which are the sex cells. In animals, these are sperm and egg cells, and in flowering plants, these are pollen and egg cells. Each gamete contains half the number of chromosomes of the body cells, so that would be 23 in human gametes. When fertilization occurs, these both combine to form the full set of chromosomes, which are a mixture of each parent's chromosome. This means the offspring, which is the child, shows genetic variation as their DNA is different. For asexual reproduction, only one parent is needed, and so there is no mixing of chromosomes and no genetic variation. The offspring is a clone of the parent. This type of reproduction occurs in bacteria cells and is much more rapid and efficient than sexual reproduction. Mitosis and meiosis are both examples of cell division, where mitosis is asexual reproduction and meiosis is sexual reproduction. Mitosis is used to make new body cells and involves one division, which produces two daughter cells which are both identical. Whereas in meiosis, this is used to make gametes and involves two divisions and results in 4 daughter cells being produced, and all 4 of these are different to each other. Next up, we have DNA, which is found in cells in the nucleus. Within the nucleus, you have structures known as chromosomes, and these are made up of strands of DNA. The DNA itself is a polymer in the form of 2 strands coiled up into a double helix shape. The coded information in the DNA is used to make proteins, as it instructs the cell to join the amino acids together in a particular order. This makes a particular protein. A gene is a small section of DNA found on a chromosome that's responsible for coding specific proteins. The genome, on the other hand, is the entire genetic material of an organism. The human genome has now been fully studied by scientists and has been useful for finding genes that cause particular inherited diseases. It's also been useful for treating inherited diseases and tracing human migration and seeing how our ancestors moved around the planet. Genes are responsible for the different characteristics within an individual, but you can have different forms of genes which are known as alleles. For example, the eye-colored gene can have one allele which gives a person brown eyes and another allele that can give a person blue eyes. Alleles exist in pairs, where each individual inherits one allele from each parent. Sometimes these are the same, but sometimes they're different. But the alleles in a pair can't both express their traits in an individual. So for example, a person can't have both blue and brown eyes. One allele can be more dominant than another. So a dominant allele is one that always expresses itself when it's present, whereas a recessive allele, which is a less dominant one, can only express itself if both alleles in the pair are recessive. If both alleles in the pair are the same, we say that they are homozygous, but if they're different, we say that they're heterozygous. The combination of the alleles present is known as the genotype, and the physical characteristics that it expresses is known as the phenotype. You can use genetic crosses and Punnet squares to find out the probabilities that offspring will have specific genotypes and phenotypes. To do this, you write the genotypes of each parent on the sides of the diagram and write the combinations in the middle. From this, you can find the probability that the offspring will have specific genotypes. So for this example, 2 heterozygous parents would have a 25% chance of having a blue-eyed child. You can use these diagrams to also find the probability that an individual will have an inherited disease. Cystic fibrosis, for example, is a disease caused by a recessive allele, and it leads to thick and sticky mucus being produced in the airwaves and pancreas. For diseases caused by recessive alleles, you can have carriers, who are individuals who have one copy of the recessive allele. This means that they can have the gene for the disease, but they don't have the disease itself. This means carriers of cystic fibrosis can have children who have the disease, even though the parents themselves don't have the disease. Polydactyly is another genetic condition, but this one's caused by a dominant allele. This is when a child is born with extra fingers or toes. You can't have carriers of polydactyly because any person with a polydactyly allele will always have the disease as its dominant. Punnet squares can also be used to show that there's a 50-50 chance of having a male or female offspring. Females have 2 X chromosomes, whereas males have 1 X and 1 Y chromosome. So when a mother and a father have a child, the Punnet square shows that there's a 50% chance the child will be a female with XX chromosomes and a 50% chance there'll be a male with XY chromosomes. Next up, we have variation, which refers to the differences in characteristics of individuals within a particular species. For example, all humans on Earth are different to each other. This could be due to genetic variation, which are the characteristics humans get from their parents due to the random mixing of DNA, and environmental variation, which is due to the conditions in which they have developed. Variation can be caused by mutations, which are random changes in the sequence of DNA. These can lead to a gene being altered, which can also lead to a different sequence of amino acids. This can lead to different proteins being made. These changes can lead to a change in the phenotype of an organism. If the mutation happens to be beneficial to the organism's survival, it could lead to the organism's species evolving. Evolution is the gradual development of the inherited characteristics of a population over time through natural selection. All organisms show variation in their phenotypes and compete with each other for resources such as food and shelter. If they have beneficial phenotypes, then they are better suited to their environment, meaning they are more likely to survive, reproduce, and pass on their beneficial genes to their offspring. This happens over many generations, and whatever the beneficial characteristic was will become more common within the species. When this happens, we say that the species is evolving. If they evolve enough, it's possible for the 2 different populations of one species to become so different in phenotype that they turn into 2 different species. This occurs when the 2 different populations change so much that they can no longer interbreed to form fertile offspring. Sometimes species can become extinct, and this is when the species has no surviving individuals. This can happen due to environmental changes, new predators or disease, competition, natural disasters, and also human activity. The theory of evolution was developed by Charles Darwin, and it wasn't accepted by many people during his time. This is because there wasn't much evidence for it. He suggested that the traits for beneficial characteristics in organisms were passed down to their offspring. This theory was only accepted later when DNA and genes were fully understood. We found out that characteristics were passed down to offspring through genes. Other evidence we had was antibiotic resistance, where some bacteria cells mutate and don't get killed by antibiotics. Natural selection causes the normal bacteria cells to be killed due to the antibiotics and the resistant ones to survive and reproduce. This results in the bacteria evolving. This means it's much harder to treat diseases caused by these resistant bacteria, and a real-life example of this is MRSA. To prevent this from happening, doctors should only prescribe antibiotics for serious bacterial illnesses, and patients should complete the full course of antibiotics to ensure all the bacteria are killed and none can survive and become resistant. Another piece of evidence for evolution are fossils, which are preserved remains or impressions of ancient organisms. They can be formed by 3 methods. One is where hard parts of an organism, such as teeth and shells, are gradually replaced by minerals. Another is when an organism dies in areas where microorganisms can't work, such as amber, tar pits, glaciers, or peat bogs. And the third way is when trace fossils are formed, which show the movement and activity of ancient organisms. This could be things like footprints in clay or rootlet traces, which show where plants' roots used to be. This also includes burrow traces, which show tunnels that animals used to travel through. But fossils only give some evidence. For example, soft parts of organisms decay, so very early life forms left very few fossils as they were mostly made of soft bodies. All living things can be classified in different ways, and these organize them into different groups. The Linnaean system was made by Carl Linnaeus and classified organisms into several stages of subdivisions. These are kingdom, phylum, class, order, family, genus, and species. Any organism can be named using the binomial system, where you just combine the genus and the species of the organism. So if these are the subdivisions for humans, the binomial name for a human will be Homo sapien. Another system that was created by Carl Woese used a 3-domain system where all organisms were divided into 3 groups. Archaea, which are primitive bacteria found in extreme conditions, bacteria, which are the more well-known bacteria that we know, and eukaryota, which include fungi, plants, animals, and protists. Next up, we have selective breeding, which is where humans choose specific plants or animals with desirable traits to breed so that they can pass down the desirable characteristics to their offspring. Examples of these desirable characteristics could be things like disease-resistant food crops, animals which produce more meat or milk, pets with a gentle nature, and decorative flowers which are large or unusual. To carry out selective breeding, you first select the best individuals. So for example, for decorative plants, this will be the ones with the biggest flowers. You then breed them, and from the offspring, you select the ones with the biggest flowers again and keep repeating the process. The population of the plants with the big flowers will increase over many generations. The main disadvantage of selective breeding is that there's a reduction in gene pool as all the individuals are closely related. This means there are less alleles present in the population, so less variation in it. This can lead to the population having genetic defects and being more susceptible to new diseases. Genetic engineering also involves creating organisms with desired characteristics, but this involves physically modifying the DNA of the organism so that it develops with the desired characteristics. This involves isolating and cutting out the gene for the desired characteristic from the human cell. So for example, if we wanted to create insulin for treating diabetes, we would cut out the gene responsible for making insulin. We then insert this into a plasmid of a bacteria cell. The plasmid is known as a vector, and you can also use a virus as a vector for the same purpose. Now you can put the plasmid back into the bacteria cell, and now the bacteria cell is able to make insulin. So you can allow them to reproduce so even more insulin can be made. This method can also be used to make genetically modified crops which are resistant to disease, insects or herbicides, or even ones that make bigger and better fruit. But there are both advantages and disadvantages to using this method. The final topic in biology is ecology, where you need to understand that an ecosystem is the interaction of a community of living organisms, which are known as the biotic parts, with the non-living parts of their environment, which are known as the abiotic parts. Biotic or living factors that can affect ecosystems include new predators, competition with members of the same species, new pathogens that cause disease, and the availability of food. Abiotic or non-living factors, on the other hand, could be things like moisture level, light intensity, temperature, carbon dioxide level, wind intensity and direction, oxygen level, and soil pH and mineral content. All organisms within a community are interdependent, which means they all rely on each other. And for a community to be stable, their population sizes need to remain fairly constant. The organisms can be adapted to their environments to help them survive better. This can include one of three types of adaptations. Structural adaptations, which involve physical characteristics such as body shape and color. So for example, polar bears have white furs to camouflage into their surroundings. Also behavioral adaptations, which are actions organisms take to survive better, such as a bear hibernating in winter. And functional adaptations, where the bodily functions of an organism help it to survive, such as camels conserving water by producing very little sweat or urine. Some bacteria known as extremophiles have adapted to live in extreme conditions such as high temperature, high pressures, and high salt concentrations. Within an ecosystem, you can have food chains that show you what an organism eats. These always start off with a producer such as plant or algae that can photosynthesize to produce its own food. Then it gets eaten by a primary consumer, then a secondary consumer, and then a tertiary consumer. Biomass, which is the total mass of living material in an organism, can be thought of as energy. This gets transferred along the food chains. The amount of biomass or energy transferred decreases after each stage, so the top of the food chain basically receives the least amount of energy. The organisms that kill other animals in a food chain for food are known as predators, and the organisms they eat is known as the prey. In a stable ecosystem, the numbers of predators and prey tend to slightly fluctuate in a repeating cycle. Next up, we have the water cycle, which shows how water recycles around the environment. It evaporates from the ocean and also plants as transpiration and condenses as it rises in the air. The water then falls to the ground as precipitation, where it can run off into the ground and go back around again. Similarly, we have the carbon cycle, which recycles the element carbon around the environment. Carbon dioxide in the air is taken in by plants through photosynthesis, where the plant uses the carbon to grow. This carbon then gets transferred down food chains to animals. When both plants and animals die, their dead material decay and get broken down by bacteria known as decomposers. All living things respire and release carbon dioxide into the air, and this includes plants, animals, and decomposers. Plants and animals that died and got buried in mud millions of years ago got turned into fossil fuels. Humans dig these fossil fuels up and combust them for energy. This releases carbon dioxide back into the air. Carbon dioxide is known as a greenhouse gas, as well as methane and water vapor. These gases trap heat radiation from the sun onto the earth. This acts as an insulating layer and causes the earth to be warm. However, due to human activities, this effect has increased. This has led to global warming, where the average temperature of the earth increases and leads to rising sea levels, flooding, and shifting ecosystems. Deforestation has had an impact on greenhouse gases, as cutting down trees has meant less carbon dioxide has been taken in by the trees and more carbon dioxide has been released due to the dead wood decaying or being burnt. Deforestation has also led to a reduction of biodiversity. This is basically the variety of different organisms on Earth or in an ecosystem. A high biodiversity is important as it keeps ecosystems stable and functioning effectively. Peat bogs also affect biodiversity. They're unique areas of land found in waterlogged and acidic areas. They consist of peat, which is made of plant material that have only partially decayed. They're important as they lock in carbon dioxide that will usually be released into the environment. They also act as habitats for many different species, so they maintain biodiversity. Human activities such as farming have destroyed peat bogs, which has enhanced global warming and reduced biodiversity. To minimize the effects on ecosystems and biodiversity, scientists have introduced different programs such as breeding programs to prevent endangered species from becoming extinct, habitat protection and regeneration, recycling to reduce pollution from waste, reduction of deforestation and carbon dioxide emissions by governments, and introducing field margins and hedgerows where farmers grow only one type of crop.